Chapter 15 of Campbell Biology, titled The Chromosomal Basis of Inheritance, can seem like a really tough chapter, but I'm here to tell you today that you only need to know one thing, and that's what this video is about. Hey guys, this is Mikey from Able Prep Academy, and on this channel we talk AP Bio content, and in today's video we're going to talk all about genetic linkage. Linkage appears in Chapter 15 of Campbell Biology, and it's also part of Unit 5 on heredity. But unlike Mendelian genetics, which can seem relatively easy for AP Bio students, linkage can be pretty tough, especially as it pertains to some of the mathematical concepts that you need to link to it. So let's clarify the concept of linkage by first investigating the Mendelian law called the law of independent assortment. The law of independent assortment is a very important law proposed by Mendel when analyzing two genes simultaneously. And to demonstrate this easily, let's imagine that you have a dihybrid individual with big A little a and big B little b as their genotype. What the law of independent assortment states is that this individual should be able to produce 4 different gamete types, one with big A big B, one with big A little b, the other with little a big B, and the last one with little a and little b. Now, I'm sure many of you guys weren't thinking too much about exactly why you're able to create these 4 gamete allele combinations, but there's a lot of science behind exactly why those gamete combinations are formed. Looking back to meiosis, let's imagine for a moment that these genes A and B are on 2 separate chromosomes. On one pair of homologous chromosomes, one of the chromosomes would have the big A and big A allele on both chromatids, and the other chromosome would have little a and little a on the other chromatid. This would also be true for gene B, where this individual would have big B and big B on one chromosome, and on its homologue, it would have the little b on both of the sister chromatids. But something cool happens during metaphase 1 of meiosis. On metaphase 1 of meiosis 1, these chromosomes would randomly align themselves along the equator of the metaphase plate, which means that you could have a combination where both the big A's and the big B's are at the top of the equator and the little A's and little B's at the bottom, or you could have the other combination where you have a big A and little b on top and little a and big B on the bottom. Now, because these two possibilities are one or the other, there's roughly 50% chance that one or the other phenomena would occur. Now, if you're to follow through with everything else that's supposed to occur in meiotic division, then you would have a 50% chance of producing gametes that are big A, big B, little b, or big A, little b, and little a and big B. But there's a catch. This is only possible if genes A and B are on 2 different chromosomes. If genes A and B were on the same chromosome, then no matter how these chromosomes would align at the metaphase plate, it would not make a single difference. Whatever allele combinations are found on one chromosome or the other would result in producing gametes with just those combinations. But we know that even when 2 genes are on the same chromosomes, you still get those different gametic combinations to occur. So the question is how? And that is where crossing over comes into play. See, when 2 genes are on the same chromosome, but they're relatively far apart, what ends up happening is that you have random crossover events that will recombine those alleles on one chromosome and the other, and you end up with what we call recombinant chromatids that would have shuffled the alleles between 2 homologous chromosomes. And this would always happen as long as there are odd numbers of crossover events occurring between the 2 loci. Even number of crossovers would probably reshuffle the alleles back into their original configurations, but whether you get an odd number or an even number is again 50-50%, meaning that you'll still get those 4 allele combinations arising in your gametes. Now, we can actually demonstrate this by looking at some data. Imagine a cross between a dihybrid individual and a double recessive individual. Because a dihybrid individual produces these 4 gamete combinations in equal parts, you would have 1/4 chance of big A big B, little a big B, and little a little b from that dihybrid parent. But the double recessive parent would only be able to contribute little a and little b, so we can simply put this parent up at the top and create a 4x1 Punnett square. Into the Punnett square, we can fill in the genotypes of the offspring. Now, if we were to actually do this with, let's say, fruit flies looking at 2 genes, then we would be able to see that when we count up the offspring, there would roughly be 25% in their representation. This, by the way, is also sometimes called a test cross. However, sometimes we get results that we don't expect to see. For example, what if we had these results? For example, we might have big A, big B, and little a, little b arising in much greater numbers than the other combinations. Well, because the double recessive individual would not be able to contribute to the changing ratios of these offspring, that means that the results that you see are being purely driven by the dihybrid parent. And to make this a little bit easier, it means that the parent is producing big A, big B, and little b gametes way more than big A, little b, and little a, big B gametes. And the question is why? And the answer was discovered by Thomas Morgan with Drosophila, and he call this linkage. You see, Morgan discovered that if two genes are on the same chromosome and if they're really close to each other, then the chances of a random crossover event occurring in between those two genes decreases. You can think of two genes as being two targets between which some event is supposed to occur. As those two targets become closer and closer and closer, some event occurring in between those two close distances become rarer. For example, imagine that you're playing beer pong.
Dad, they're in high school.
Oh, sorry. Imagine that you close your eyes and throw your pencil at your wall. The chance of that pencil hitting that wall at some point is pretty high. However, if you're trying to aim for that corner where that one wall meets the other, well, that's going to be a lot harder if you close your eyes and throw your pencil randomly. And because of this, if the parental chromosomes had big A and big B and little b on the same chromosomes, then the chance of recombining those alleles into big A and little a and big B become rarer and rarer and rarer. And as a result, you don't see this 1-to-1-to-1-to-1 ratio, but rather a ratio that seems completely skewed. And this is one of those examples where the genes are not independently assorting with respect to one another. Now, there are two major math applications that arise from this concept. One is chi-squared analysis and the other is linkage mapping. And for linkage mapping, which is important for this test, you can click on the video card at the end of this video to figure out how to do that. But for now, this has been Mikey with Avo Prep Academy. Don't forget to like and subscribe, and we'll see you in that video in just a few minutes.